Now that we've considered proximal distal patterning and anterior posterior patterning, now we're going to turn our attention in this second podcast to dorsal ventral patterning. This is going to be a role that is played by the ectoderm. And so we're going to look at the different factors that are expressed in different positions of the ectoderm and how those chemical signals impact the development of the underlying mesoderm. Now, some of these studies have been done to really help us understand when is the mesoderm determined and when is it specified. And so a lot of these right-left swapping studies have done, and you might think that if you swap right and left, the wings will be formed backwards. And there's other studies we'll do with forelimbs and hind limbs. And so you look at those structures, and when you swap, proximal structures are normal because of the time they spent being interpreted. But distal structures, the hand and foot, for instance, if we did those are reversed. So what we see that's important is that in the dorsal ectoderm, and dorsal meaning anything dorsal of the ectodermal ridge, is going to express a factor that is called radical fringe. Now, a lot of these were identified in Drosophila, so that's why they have these sort of interesting names. So radical fringe is going to dictate dorsal ectoderm, and it's going to have influence on the dorsal mesoderm that immediately underlies it. So as we continue to look on, we see that Wnt, this particular Wnt 7a, this is going to be expressed in dorsal ectoderm as well. And the Wnt 7a is going to induce the underlying mesoderm to express LMX1. Now, this is going to be a homeotic gene, and we'll get to those in a minute, but those are transcription factors, and that's going to drive the expression of dorsal structures. Now, as we mentioned before, we can overexpress materials in the limb, or we can do knockouts where we prevent the expression of certain genes. And in the case of Wnt 7a, if we knock out the expression of Wnt 7a in our dorsal ectoderm, then we get somewhat of a mirror image in the pattern, and we get double ventral. So we have ventral structures on both sides. So the dorsal has been eliminated. Now, we look at LMX1, and we know that LMX1 is going to be a dorsal gene. So if we overexpress LMX1 in that mesenchyme, in this case in the ventral mesenchyme, then we get a double dorsal. So LMX1 is directing dorsal structures. Here's the interesting thing. If we knock out LMX1, it causes our dorsal structures to be absent. So dorsalization doesn't happen when we knock out LMX1, and that's what we would expect. However, no double ventral phenotype occurs. So you don't get dorsal and you don't get ventral. You get something in between, and I've never seen a picture of what this in between looks like, but you don't get a double ventral. Now, when we look at our ventral ectoderm, so far we have seen that we've got radical fringe and Wnt 7a expressed in the dorsal ectoderm. That's going to lead to the expression of LMX1 in our dorsal mesenchyme. Now, if we look at the ventral side, we look at the ectoderm, we see engrailed, and this is going to be a protein produced there. And if we knock out engrailed, then what happens is Wnt 7a is expressed in both dorsal and ventral, and we get a double dorsal limb. So it appears that Wnt 7a might be the default pathway, and engrailed might be a means of inhibiting Wnt 7a to allow for the expression of ventralized structures. So here we see Wnt 7a expressed in our dorsal ectoderm. Here we see engrailed expressed in our ventral, and here we see the expression of LMX1. We haven't identified factors yet that occur in this ventral mesenchyme, but this is our dorsal ventral patterning, seemingly expressed and directed by inductive forces coming from the ectoderm. Now we can fill in this portion of our pattern, and that's going to be our dorsal ventral axis, and this is going to have an impact on all of the developing mesenchyme. And you can see there's nothing you can really tease out and say, this is specifically what's happening here. This is, if this, then this. Well, you know, it's all working together. So really, when you look at the three pathways and the major players, well, we've got to go with FGF4, which certainly is the initiation and the elongation. Sonic Hedgehog, which is playing a role possibly in the initiation, but certainly digit identity and number. And then Wnt 7a with the dorsal and ventral patterning. So all three of these interrelate to give us a normally developed limb. Now, how do we then rationalize if these are occurring in both hind limbs and forelimbs? How do we end up with wings and legs as opposed to all of them being the same? If those are the only chemicals that are produced, how is anything any different? And so in this case, they transplanted limb, apical ectodermal ridge, into the AER, transplanted limb ZPA into the wing ZPA to see if they could decipher the signals that are maybe directing limb structures, wing structures from leg structures. Well, when you do all these experiments, it's more than just the signals, but how does the mesenchyme and when does the mesenchyme interpret those signals that allows things to happen? So here, for instance, is a pretty powerful experiment. Here we have wing tissue and leg tissue in the chick. If we allow the mesenchyme to develop for a specific length of time, this is a fairly good size bud, and we take some of this proximal mesenchyme that's been under the influence for the longest period of time, transplant this leg mesenchyme into the progress zone, allow it to develop, and what you see is we have normal wing structures until we get to the distal end from this progress zone region that's going to be under that influence the longest. Now we see distal structures that aren't wing at all, but actually leg structures. So again, we don't understand all of the signals, but we do know that the amount of time the mesenchyme is under the influence in a particular location has a great impact on the different elements of whether we're going to form a limb, whether it's going to be a wing, whether it's going to be an arm. So this is a very simplistic flowchart of what we see and how we actually direct that. And you know, it's more than just that. It's such a powerful thing to look at. How does the mesenchyme know? And this whole idea of pre-patterning becomes sort of a very esoteric thing. But we see that the determination and specification happens fairly early. And it appears that it's going to be independent of the ZPA for this anterior-posterior axis formation. Certainly when we transplanted the leg tissue into the wing progress zone, well, we still got leg tissue, but they were distal. They weren't proximal structures. So again, we're sort of getting glimpses of the big jigsaw puzzle, but not everything together. So here's an experiment that we did. So you take the leg bud, you cut it off, you dissociate and scramble up the mesodermal cells. You don't kill them. And you pack them back into this ectodermal cup, put it back on the embryo, and you let it develop. Well, when you do that, certainly you've scrambled all the patterning that was going on in these proximal structures, time in the progress zone, etc. But once you put it all together, wow, look at these distal structures. You get toes, and fairly normal looking toes. So the cells that were specified early on, they lost their positional signaling. But the cells, regardless of where they were when they came under the influence of that progress zone later, that progress zone was able to redirect them back into normal tissue. In this case, it's leg mesoderm. It became distal leg tissues. If you were to take a wing bud, scramble it, put it back in with a leg ectoderm in a leg position, you would still grow wing tissue because of this pre-patterning, and the pre-patterning directing either a leg or a wing structure. Now, the last thing I want to cover in this unit and in this course are these transcription factors, these DNA binding factors that are called homeotic genes. Now, homeotic stems from this domain that was called the homeobox. This homeobox is a helix loop, and back in the day we used to call it a helix loop helix motif. And it's going to be 60 amino acids that can actually bind in the grooves of DNA, the major and the minor grooves, and that's how it's going to be directing where, in fact, it interacts with the DNA. Here you can see a homeotic gene, and you can see the helix, loop helix, and you can see how this helix fits right into that groove of the DNA. Then we're going to have other DNA portions that interact and open the DNA so that it can be transcribed in a certain position, and those gene products be activated. Now, when we look at the homeobox motif, if you will, here's the homeodomain, the homeobox. When we look at this in various species and various positions, here we can see that we've got a couple of Drosophila genes. Labial is one, deformed is one, Antennapedia is one, and we compare them to all other homeotic genes in this region. You can see there is a tremendous amount of conservation. Now, HOX1b, HOXB4, HOXB7, HOXB9, you might think that these are very related species to Drosophila to have that much in common. Look at this Antennapedia and HOXB7. It's almost the identical homeodomain, and these are amino acids. This is not nucleic acid code. These are the 60 amino acids. HOXB7 is a mammalian gene. This is found in mice. And so think of the evolutionary distance between the invertebrate Drosophila and a mouse, and you can see how conserved these sequences are. And so there must be something significant going on with these homeodomains and these homeotic genes. Well, again, as I mentioned, Drosophila has a lot to do with it because it was the first organism where we identified these HOX genes through various mutations. And actually the first mutation, they saw fruit flies walking around with legs growing out of their head. Instead of antenna, they had legs. And so the first homeotic gene identified was termed Antennapedia. That was the term for the mutation. And they did backward genetics then to find the mutation that had led to the formation of the legs instead of antenna. And they were just quite puzzled. Well, what they found is they looked at these homeotic genes, and as we look at Drosophila on this single chromosome, the genes are expressed in a 5' to 3' pattern that mimics their anterior-posterior patterning in the embryo. We can see that labial, the most 5', it's the most anterior. Abdominal B, it's the most 3' on the chromosome, and it's the most posterior. And as we look at mouse homeotic genes and even human homeotic genes, we can see that same chromosomal pattern is going to be re-expressed in positional information body plan of the organism. Now, Drosophila had a single chromosome, but as we went through evolutionary history, there were duplications of these clusters of chromosomes. And so as we get to vertebrates, now we see that there are 4 clusters and 4 different chromosomes that have these homeotic genes. And if we look at the plan, here is the chromosome from Drosophila. And if we look at the 4 clusters of homeotic genes, and they just simply number them 1, 2, 3, and 4, we can see, again, the 5' to 3' positional information conserved on the chromosome, and that relates to its expression in the actual organism. And again, this is through all of our vertebrates. We can look at fish, salamanders, chicken, rabbit, and as we saw the mammals, we express those as well. What does that really have to do with limb development, you might say? Sure, that's a nice body plan. Well, we see the HOX genes expressed in a positional information in the wing as well. But when we look at the HOX genes in vertebrate development, the best example is the vertebral column. This is actually the best. So here is the hypothetical model that we see. Again, we have 5' genes, 3' genes. We would expect to see this positional information as it relates to our vertebral column. And in fact, it's not just that gene 1 is expressed here, gene 2 is only expressed here, but we see sort of a nesting of expression. So that if we're going to develop an anterior structure, it's going to be directed simply by homeotic gene 1. But a more posterior structure is going to be expressed by these 2 working together, even more posterior, 3 working together, and the most posterior in this case working together, all 4. So that's this nested hypothetical model of how the Hox genes direct positional information in an anterior-posterior pattern. And in a mouse embryo, we see that in almost every cluster. Here's HOXB1 expression. It begins in the cervical region, and even though it's very light through here, we can see most of that dark staining occurring throughout the remainder of the embryo. As we move backward and look at B4, more 3', it's not expressed as high as B1. It starts a little bit lower, but it's expressed throughout. B9, much lower and expressed throughout. And actually, when we put these together and look in our vertebral column, this is anterior and here is posterior, we can see that the 1 clusters, the 3, 4, 5, 6, all the way down to 13, does show this nested pattern from this hypothetical model that we proposed. It is conserved in our vertebral column. Now, doing the knockout experiments and stuff, we haven't done those quite so much, but in some instances we have, so that if we look at Hox C8, the C cluster and the 8th gene, we look at the normal vertebrae. We have the 13th thoracic and the 1st lumbar vertebrae. If we knock out just the Hox C8 gene, this is what we get. We get a 14th thoracic vertebrae, and we move the lumbar, the 1st lumbar, down by 1. So we have changed the positional information of this single vertebrae. We still have vertebrae, we just changed its identity based on this anterior-posterior patterning. All right, so that's how we're working, and that's how these homeotic genes work with this positional information. Now, let's look at the limb, because we have these Hox genes expressed in the developing limb. So let's look at a few rules that we have of how the Hox genes might be assisting in the morphogenetic patterning of the limb. So remember, we have 4 clusters, A, B, C, and D. And so as we look at Hox A and Hox D, these are both expressed in both the forelimb and the hind limb. Now, for humans, we're talking about arms and legs. Hox B, you only see that cluster expressed in the forelimb, and Hox C only expressed in the hind limb. Okay, again, we've got some conservation going on with A and D, but we have some specificity with B and with C. So as we continue to look, again, we are going to see that as the limb is moving and as the limb is growing, especially with the progress zone, we're going to see that the Hox genes really are going to be a record of that positional information. But it's not something that is going to be very static. It's going to be dynamic. So as the limb lengthens, so is the difference in expression of these Hox genes. Because you can't start out, if you imagine a real small limb bud, well, you can't have all the pattern there because we haven't made much of what's going to come with that progress zone and the growing limb. So we're going to see a pattern, and again, this might be influenced by the time of the mesoderm in the progress zone itself or the distance away from the AER, depending on which homeotic gene is to be expressed. But here we see a pattern of expression in the limbs, and again, A and D are expressed in both. So we see the A cluster, we see Hox A9. It is going to be expressed throughout the entirety of the limb. But once we get into the middle portion, we see that Hox 10 and 11 is added. And by the time we get to the end, we see Hox 12 and 13 are added. So 9 appears to be more proximal, 13 appears more distal, and then you have this nested progression like we saw in the vertebrae. For Hox D, we see something similar, but it's not this really nice linear pattern. It's more offset, more at this angle. But again, we can see 9 is more proximal, 13 is going to be the most distal with a nested pattern between. So basically that same nested pattern we saw in the vertebral column, it does exist in both our forelimbs and in our hind limbs. Now, like we did with FGFs and sonic hedgehogs, we try to manipulate this. We try to knock these things out and say, okay, well, what is the result of what's going on? What do the Hox genes have to do with anything as far as patterning in the limb? Well, whenever we do our ZPA experiment, whenever we get duplication of the digits, guess what? The Hox genes are going to mimic that as well. So again, we see sonic hedgehog coming on when we do the ZPAs. or even when we do the FGFs, now we also see the Hox genes are also going to be induced to form. So is it the Hox genes as a consequence of having a new ZPA that actually directs digit number and digit identity? Okay, we still have to do more experiments to sort of find this out. So let's look at some of these. And again, it's not crystal clear, but if we knock out A and D, remember these are expressed in all genes. 11 is going to be something somewhat in the middle. Well, when we do that, we see no radius in ulna. 13, overexpression. Right? This is one of the more distal ones. Okay. Radius and ulna are going to be shorter. Now that I can't wrap my brain around because we should be thinking of phalanges at this point. But 13 overexpression may actually spill into these more proximal regions, and so it may influence those, and that might be why the radius and ulna are shorter. 13 mutation. So if we mutate D, remember that's expressed everywhere, we get polydactyly or fusion of the digits. We talked about that in class today. But that's just a multiplication of the digits. And again, it's over and above what would normally be there. Hox A13 mutation. Mutation, are we overexpressing, underexpressing, changing it, knocking it out? They're not clear in some of these studies in the book. But if we mutate Hox A13, anterior, posterior digits are reduced in size. So basically, the bottom line is when we manipulate only the Hox genes, we are conferring a different phenotype on our organism. Now, this is a summary of our knockout experiments. The reason that we have this minus/minus is we have 2 alleles, remember, for each one of these. And if we knock out both alleles so that we can't express this, then if we knock out Hox 9, only our most proximal humerus in this case doesn't form. Knock out both of the 11s. We're going to shorten the radius and the ulna in some of these carpals. If we knock out 13, a lot of our distal structures aren't formed. The fact that we still have some, it's a little puzzling. But certainly the distal structures are under the regulation of 13. Some of the middle structures are under the expression of 11. And 9 is directed here by the most proximal structures.